I'll ask you to unshare your screen and we'll move to the final talk in this session. Ţuţu is going to talk about dynamic reconfiguration via typed modalities. Ionuţ is there? Yeah, perfect.
Thank you, Rosemary. Let me just check if the presentation works well. So I guess you can see my slides now.
Yes, go ahead.
Okay, thanks. So this presentation is about model logics and also just a tiny bit about biological models and mathematical models used in biology. It's also about the way we use modalities to model and reason about reconfigurable systems, and about some of the difficulties that we've encountered in this process and some ways we found to overcome them. And it's joint work with Claudia Chirizza from the University of Edinburgh and with Joseph Yadeiro from the University of Dundee. So in this talk, we'll get to see a new kind of model logic, which, unlike conventional formalisms, uses typed modalities. And we'll get to see why this sort of upgrade of the familiar notion of modality is necessary. Then in the second part of the talk, I'll illustrate the methodology that we use for modeling and reasoning about a particular class of reconfigurable systems, ones which we call dynamic networks of interactions, or DNI for short. Good, so let's start with the motivations. Dynamic networks of interactions are networks whose configurations change dynamically as a result of interactions that take place between their components. Those components vary a lot in concrete cases. They can be agents, actors, all sorts of artifacts, and so on. Formally, the networks are labeled transition systems or cryptic structures, where states correspond to network configurations. So they have some kind of inner structure that is specific to the application domain of the network. And transitions correspond to reconfigurations. They are triggered by interactions, as I just said, and are also specific to the application domain. Let's look at an example to understand what I mean by being specific to the application domain. Membrane budding is a biological process that takes place within cells. It involves the transport of some molecules, which we call cargo molecules, from one organelle to another. In order for this transport to be possible, cargo molecules need to be enclosed in carrier vesicles, which is what we see on the screen right now. So this black circle represents the membrane of the vesicle. Inside it, we have three teardrops depicted in blue, which represent cargo molecules. And outside it, we have 5 yellow squares that represent coating proteins, which facilitated the formation of the vesicle at the first place. So we'll get to this in the next slides. And at the bottom of the screen, what we see as this black line is meant to represent the membrane of the organelle from which the 3 cargo molecules originate. Now, membrane budding is a process that explains how these vesicles are formed through the deformation of the membrane of the source organelle, that is the black line that we see on the screen, under the joint action of some coating proteins, the yellow squares. And the process unfolds as follows. We start with coating proteins and organelles that coexist in a cell. Once the protein gets close enough to the membrane of an organelle, it can bind to the membrane of that organelle, which we depict like this. And when cargo molecules, which initially reside inside organelles, get close enough to the binding site, they can be captured by proteins. These binding and perhaps capturing stages repeat until a coat of proteins is formed on the surface of the organelle. And when that coat is complete, which means that the joint force of its proteins is strong enough to deform the membrane of the organelle, a bud is formed. So a bud is just a deformation of the membrane under the action of the coat proteins. Then the bud detaches from the membrane. We get a vesicle, which can travel to another organelle to deliver its cargo. So that's membrane budding in a nutshell. Suppose now we attempt to model this process using standard model logic. And we look for now only at the binding of proteins to organelles. One possibility would be to write a sentence of this form. So for all proteins P and all organelles O, if P is close enough to O, and if it's unbound, so it's not bound to an existing organelle, then once binding occurs, there exists some code C, which contains the protein that we've just bound, and has the same membrane as the membrane of the organelle. So in other words, this coat that is formed on top, on the surface of the organelle. Under the usual semantics of quantified modal logic, this turns out to be problematic, because given that P and O are free variables, and that there's no connection between P, O, and this modality bind that we use here, it follows that we may have situation where we have, say, a protein P, like the yellow square that we depict here on the left, which is close enough to two distinct organelles, depicted by the top and bottom black lines in these dotted circles. So according to this axiomatization, the protein P would end up being simultaneously bound to both organelles, which is a biological inconsistency. A protein can only be bound to one organelle at a given time. Okay, so what we would like to have instead are two reconfigurations, one binding the protein to the top organelle and another one binding the protein to the bottom organelle, but this is not in agreement with the ordinary semantics of modal logic. Therefore, we need to revisit the notion of reconfiguration. Traditionally, reconfigurations are plain transitions between configurations. Here we depict configurations by dotted circles and components of configurations by blue disks inside the configurations. But what we need is to identify what are the actants that actually trigger the reconfiguration. So, for example, binary configuration is triggered by a protein and an organelle that are sufficiently close to each other. And we also need to identify the specific outcomes of a reconfiguration, like the generation of a new coat once a protein gets bound to an organelle. So the conclusion is that in this setting of dynamic networks of interactions, reconfigurations are not mere links between configurations, but there are some kind of hyperlinks between some components in the source configuration, that these would be the triggers, and components in the target configuration, which would be the outcomes of the reconfiguration. Okay, so let's see how we get to formalize all this. We follow the usual universal logic approach, which means that first we define the signatures of the new logic, then the models, sentences, and the satisfaction relation, and so on. The signatures of this logic mix together features that are specific to first order logic on one hand, and features that are specific to modal logic on another hand. So first we have first order signature delta of global symbols. Global meaning that their interpretations do not depend on configurations or on the existence of configurations in any way. Then we have another first order signature omega of local symbols whose interpretations do depend on configurations. So, for example, definitions of what it means, what is the structure of codes and so on, those would reside in delta. Whereas definitions like what is the membrane of a code, what is the membrane of a bud, and so on, those would be local. Some symbol may appear both in delta and in omega. So they are shared between the global signature and the local signature. So we call them shared, and their interpretations are required to be the same across configurations. So all this is reminiscent of quantified modal logic. Then we have nominals, which are the same as the nominals in hybrid logics, meaning that we use them as names for configurations. And lastly, and most importantly for us, we have a collection lambda of typed modalities. Each of these modalities has three components. It has a name like bind, merge, capture, and so on. It has an inner type, which indicates the triggers of that sort of reconfiguration, like the triggers of a binary configuration would be the protein and an organelle. And it also has an outer type, which indicates the outcomes of that specific kind of reconfiguration. Okay, now for the models or networks of interactions, as we call them, these are generalized Kripke models. I won't go into details, but I will be happy to discuss them further if there's time at the end. Suffice to say that each of these networks comprises a global first-order structure D, a Kripke frame W, and some local first-order structures M. So it's a mix of Kripke structures and first-order structures, which are somehow coherent according to some properties that they need to satisfy. Now, for the sentences. These are built in typical inductive fashion. We have equational atoms which state that the evaluation of the terms T1 and T2 here are the same. We have relational atoms which state that the evaluation of some terms T enjoy a property that is given by this predicate pi. We have local membership atoms saying that the evaluation of a term T is local, meaning that it belongs to a configuration that we consider. Nominal sentences exactly as in hybrid logic. So they state that the configuration is the same as the configuration that is denoted by this nominal I. Then Boolean connectives as in first order logic. Also local satisfaction operators, exactly as in hybrid model logics. And the most important for us, this upgraded form of model operator that is based on type modalities. So this sentence states that a rho holds in all configurations that are obtained along some interaction alpha, which what we have in the middle here in the middle of the model operator, that has T, a tuple of terms, as triggers, and Z, which is a tuple of variables, as outcomes. We'll get to see an example of this very soon. And lastly, we have universal quantifiers. The only thing to note here is that we can quantify both over components through what we write as X and over configurations through what we write as K. Now, of course, there are many more operators that we could consider, like possibility operators, existential quantifiers, actualist quantifiers, the store operator that exists typically in hybrid logics, and so on. But these are all derived in the sense that they can be expressed in terms of the other core operators that we've already seen on previous slides. Let's put this logic to use and see how memory embedding could be specified, but properly this time in the sense that it matches the biological interpretation of things. We could have written a large monolithic specification of memory embedding that contains everything. So all first order symbols that we need, all type modalities, all the actions describing how reconfigurations take place, and so on. But this turned out to be quite impractical for verification purposes. So what we decided to do instead was to structure the specification into modules. We have 6 core modules, which are depicted on this slide in red. Each of them deals with a specific stage of the memory embedding process. So for example, we have init, which deals with initiality constraints, bind, which deals with sufficient conditions and with the effects of bind reconfigurations, merge for merge reconfigurations, and so on. All of these modules import a base specification where we axiomatize network components. So what it means for a component to be an organelle, a protein, cargo molecule, and so on. And finally, the specification memory embedding at the bottom of the screen in the blue box is simply the union of the 6 core modules. So if we take a look inside one of the modules, this would be part of the axiomatization of binding. The first sentence, for example, reads as follows. For all local proteins P and all organelles O, binding P to O is possible, and it produces a code which we write as Z, if and only if P is close enough to O and it's unbound. So this is quite similar to what we've seen at the beginning, only that this time we avoid any sort of biological inconsistency. So the first one, this sentence that we just read, would be the progress axiom, telling us when binding is possible. The second sentence captures the effect of bind. So it's telling us that once binding takes place, the code that we obtain as an outcome is a code that consists only of the protein P that was just bound to the organelle. And there are several other axioms in this module. Okay. Now, in the last few slides, I would like to briefly illustrate the technique that we use for checking properties of dynamic networks of interactions. There's currently no dedicated proof system for DNI. So what we do is we relate DNI to other more mature and, let's say, better understood logical systems. And we borrow whatever proof systems and perhaps tools have already been developed for those logics. But whenever we do this sort of, we develop this sort of relationships, we need to make sure that they are conservative, which means that the outcome of any proof effort in that other logic, which is more mature, can be reflected back in some meaningful way to DNI. In our case, we chose to relate DNI to partial first order logic. That's because it was one of the most natural encodings to do. So the encoding takes into account both the syntax and the semantics of DNI. With respect to the syntax, any DNI signature Xi is encoded in a covariant manner as a theory representation in partial first order logic. And every DNI sentence is also encoded as a partial first order logic sentence, again, in a covariant manner. But usually this sentence in partial first order logic, it's much longer and more convoluted than the DNI sentence. And on the other hand, for models, the encoding is contravariant, meaning that for every relevant first order signature, we are able to recover or extract somehow a network. Now, the crucial property of this encoding is, as I mentioned, conservativity, which means that whenever we translate a proof goal from DNI to PFOL, so to partial first order logic, and we get to solve that proof goals using tools that are specific to PFOL, the result, which could be, say, proved or disproved, is also valid for the original DNI proof goal. So we can state something about the original problem. We have used this technique to perform a preliminary analysis of memory embedding using an experimental tool called SPEX. Suppose we verify that memory embedding, as we have axiomatized it, can indeed transfer a cargo molecule, say X, from a source organelle, which we write as S, to some target organelle, which we write as T. This is a rather complex property, rather long to write, but it's formalized as a DNI sentence like the one that we see at the bottom of the screen in the blue box, of course, with some ellipses there for the most complex part of it. This sentence makes use of all the modalities that we have defined for memory embedding, so it needs to go through all the stages of the process. And it turns out to be much too complex to be tackled directly using automatic tools and in some reasonable time. So we decided to exploit once more the modularity of the specification of memory embedding and to introduce some intermediate goals or lemmas, if you'd like. These goals correspond to the question marks that we see on the screen. So we have one for each type of reconfiguration, one for bind, merge, capture, and so on. We then processed all those goals using SPEX to produce appropriate translations into PFOL. So we moved from DNI to partial first order logic. Then we used HETS, the heterogeneous toolset, together with some automated theory improvers for first order logic like SPAS, Vampire, and E. So we've used a combination of them, but in principle, anyone would suffice now. So we use them to discharge all these proof obligations. So now, taking modularization into account, the entire process takes only a few seconds to complete. All right, so to conclude, this talk was about type modalities and about the way they may be useful in modeling and reasoning about reconfigurable systems. We have seen a verification technique that involves doing proofs by translation. But there's obviously much more to be done here. So in particular, there is indeed the need for dedicated proof theoretic support, which perhaps our hope is more efficient and convenient to use than relying on general first order theory improvers. And there are several other further research directions, which I'd be happy to discuss, particularly in relation to bigraphs. But I guess that would be all for today. So thanks a lot.
Thank you, Ionuţ. Thank you very much. Has anybody got any questions? Are the translations automatic, or how automated are they?
Yes, now they can be automated. Initially, when we submitted the paper, most of them were done by hand. In the meantime, we had a bit of time to work on this tool called SPEX, which does which is really experimental at this stage, and all it does is just parse DNI specifications and attempts to translate them into heads input, which can then be further parsed and translated to input specific to vampire, sparse, and so on.
Okay. Okay.
I would have a question, sorry.
Go ahead, Rolf.
I would have a question, sorry.
Go ahead.
Hi, Ionuţ, that's Rolf speaking here. Ionuţ, my question concerns equivalence of models. So now you have this typed modalities. So I wonder whether it's very straightforward or has some difficulties to define a bisimulation equivalence, for instance, for your models.
So this is very early work. There is some very early work on that. So I would say for now, as far as I've looked into it, it's not that straightforward. Because this can't be simply reduced to ordinary model logic. So there are some complications that arise from the use of mixing first order logic with these model operators.
Okay, thank you.
Thank you also.
Okay, thank you. Anyone else got any burning questions? No? Okay, thank you very much. I'd like to thank Ionuţ again and to thank the 4 speakers in the session. So thank you. A round of applause by myself. And that concludes this session. So thank you for your participation, and the coffee break is now on. So you can go along to the WonderMe URL, which has just been put into the chat, and I'm sure there'll be more discussion and questions there. Thank you very much.
Thank you. Bye-bye.
Bye.